My name is Fabia Battistuzzi. I'm an assistant professor at Auckland University. I've been at Auckland for 5 years now, and my background is in evolutionary genomics. So I started working with evolution of prokaryotes, bacteria and archaea. I'm one of the big areas of research in my lab is evolution of early life, but later on during my postdoc, I joined a group that does evolutionary medicine, and that's where this work started, and I'm continuing it now, basically looking at genome sequences for pathogens, in particular the pathogens of malaria, to see basically what kind of information we can get in terms of not really the epidemiology of the disease, that is something that as of right now for malaria we kind of know a lot about, but the history of the disease and how we can use the genomic information to possibly predict future changes such as drug resistance and these kind of things. So that's where this talk is coming from. Now, obviously, the whole idea behind working with malaria is that it's one of the 6 major infectious diseases. This is a relatively old report from the World Health Organization, but the numbers have not changed much. I think now we are up to about 26% of worldwide deaths are caused by infectious diseases, and the vast majority of the deaths are from 6 of them. And malaria is one of them. And if we look at how these diseases are causing deaths in different group ages, malaria, if you look at the under the age of 5 group versus the over age of 5, malaria is one of the ones that has the biggest ratio there. So it affects predominantly children. It causes death predominantly in children. Adults, usually they can handle it a little bit better, mostly because of the type of drugs that are involved in treating it. And of course, the geographic distribution. There are different pathogens for malaria, but some of them, one particular type is mostly prevalent in Africa, which is Plasmodium falciparum, which is the one that we know the most about, but then there is another major pathogen, which is Plasmodium vivax, that is prevalent mostly in South America, and then the 2 pathogens overlap in Southeast Asia, where basically both of them are present at the same time. Now, if you're keeping an eye on the news, periodically news come up discussing basically what we are doing with trying to treat, but also trying to prevent malaria and trying to decrease the number of cases that happen in a year. What this map is showing you basically are the countries that are still considered endemic for malaria in blue and in 2016, and the countries that were endemic in 2000 and are not endemic anymore for malaria in 2016. So there is some improvement with the strategies that have been implemented. Things are slowly getting better, but there is still a large majority of countries that are still having very, very big issues for a number of reasons. Part of them is, like I said, the epidemiology side of the disease we know. The genetics of the disease is still a little more unknown, but there are also some issues in implementing policies, making sure the drugs are available on a larger scale and in a financially sustainable way. So that there are some other issues that come into play into this whole problem. Now, the reason why I have this slide up, this is just basically showing you where the financing for malaria research is coming from. There are multiple, of course, pots of money, but the endemic countries, the vast majority of them are focusing basically on developing health systems. So that means, basically policies, prevention, treatments, these kind of things, and not so much on prevention. The The Global Fund is the one that is funding the majority of the prevention strategies, which means basically providing mosquito nets, providing some sort of vector control, so on and so forth. All of these strategies, treatments and prevention specifically, are helping in reducing the incidence of malaria. But because most of the endemic countries are focusing on the health system side, which is basically curing the disease but not preventing it, that's where the barrier is in changing these countries from being endemic for malaria to being non-endemic. And the way they define endemic, by the way, is if there are no cases of direct transmissions of malaria within that country for 3 years in a row, then the country is considered being non-endemic for malaria anymore. Now, another way of looking at this is looking at what kind of research is being funded, and a lot of it, as you can see here, throughout the years, not much has changed. A lot of it is basically drug development, which again is not prevention. It's great to have, but it's not prevention. Another part, of course, is the vaccines, which is the red part, and then we have the blue part, which is basic research. You put these three together, these three types of sources of information, if you will, together, and you start getting a good handle on this pathogen and on the disease itself. Now, drugs and vaccines, they are obviously developed based on information that we get from basic research, which is usually information on that is based on genomes. And so that's where the connection between the clinical epidemiological side connects with the fundamental research, because that's where the genomic aspect comes in, and that's basically where I come into this field. So, if we look at malaria, like I mentioned, there are a few different species, and we'll talk about it a little bit more in a minute, but there are a few different species that cause the disease. And within the evolutionary history of these pathogens, the Plasmodium genus, which is the agent of malaria, is a member of a larger phylum, which is the Apicomplexa phylum, which is a very unique phylum. They are unicellular eukaryotes, they're protozoans, and they're very unique because all the species that we know of Apicomplexa for now, they are all pathogenic. Not all of humans, but of other animals as well, some of them of plants, some of other protozoa, but all of them are pathogenic. So this is a very unique phylum from our perspective because they have a very strong pathogenicity signature in their genome that is present. And that's kind of what we are after. We are trying to figure out what makes them so good at being pathogens. There are a couple of things that are unique about this phylum that are beyond the lifestyle. For example, they have an organelle that is unique. It's the apicoplast. The apicoplast actually is it used to be a chloroplast organelle, so it used to be a photosynthetic organelle, but it degenerated, it lost its photosynthetic ability, and now it's an organelle that seems to be involved somehow in the pathogenicity of this phylum, although we don't quite understand how yet. It is an interesting organelle, though, because it's one of the potential targets that are being used for drug developments. Because if you think about it, when you have a pathogen, for example, if I get malaria, well, the pathogen of malaria is a eukaryote like I am, which means that any drug that I take is going to hurt a eukaryotic cell, whether it's the cell of the pathogen or my own cells. And so you get into the whole issue of drug toxicity and so on. But if you can have a drug that targets this apicoplast specifically, the apicoplast is unique to the Plasmodium. That would lower drug toxicity a lot because it wouldn't hurt my own cells. So there is a whole subfield that is looking into specifically trying to target the apicoplast. It's a different organelle to target, unfortunately, because of its evolutionary history. Because it used to be a chloroplast, it's actually bounded by 4 membranes, and so getting a drug inside the apicoplast is very difficult because it has to pass basically 4 layers of barriers. But let's say that they're working on it and see how it works. Okay, so like I said, the Apicomplexa is a very big phylum, estimated to have between 1.2 and 10 million species. So it's a very, very, very large phylum. We know maybe a few hundreds, so we really don't know much about this phylum. We are sequencing it a lot because of its pathogenic nature. But there is really not much that we know. Even going down to the actual phylogeny, so the actual relation of the different groups to each other, we are not exactly sure how they are related to each other, which means we don't know who the ancestor was, we don't know how these different characters have evolved, which is a problem if we are trying to understand the evolution of a particular disease. We do know that the vast majority of these pathogens have complex life cycles, including malaria, of course. They can infect both vertebrates and invertebrates, and in most cases the invertebrate is the vector, so it's kind of a temporary stage in a sense in which sexual reproduction happens, and then from the vector they move into the final host, which in general is a vertebrate where asexual reproduction happens. So if you look at this phylogenetic tree of Apicomplexa, Plasmodium is this little teeny tiny twig up there, and if you zoom in a little bit more into Plasmodium, into this group here, you start expanding the Plasmodium group and you have multiple species. At the time that this tree was built, it was 6 years ago, and we had only 6 fully sequenced genomes of Plasmodia, and now we have like 17. So we're getting more and more information. So within the Apicomplexa, like I said, we have a few hundreds of species that are known, but only 24 have been fully sequenced. Of these 24, we actually have multiple strains of the same species, which helps us in understanding differences in, for example, geographic distribution. So we have a number of them. Without getting into the details, the part that we are most interested in are the 17 Plasmodia. For P. falciparum, which is the one that I mentioned, that is mostly prevalently present in Africa, sub-Saharan Africa, we have 2 strains fully sequenced, and at least another 5 strains that are at the stage of scaffolds. So basically, they're mostly sequenced; they're not fully assembled yet. For P. vivax, officially we have 2 strains that are deposited in the databases, but we actually have another 3 strains that are almost ready to come out in the pipeline. And then we have a number of non-human primate malarias, a number of rodent malarias, bird malarias, so on and so forth. So the vast majority of the information that we have from the Apicomplexa is from the Plasmodium genus. And because of this information, we have been able to do a number of different studies that include evolution of the species and evolution of the populations, and that's how we are starting to understand the differences, the geographic differences of the various strains of Plasmodium. And at the species level, we're trying to figure out basically how old the disease is. The reason why we are interested in understanding how old the disease is because it allows us to sort of have a prediction of how much it will change in the future based on how much it has changed in the past. And so if we can get this sort of rate of evolution figured out, we can try to get a sense for how likely it is that, for example, it will become resistant to a new drug because it can accumulate mutations that will change its genome. So just quick overview in case not everybody is familiar with the life cycle of Plasmodium. The mosquito is the invertebrate vector, as I mentioned. Basically what happens is that a female mosquito takes a blood meal from an infected human. Unknowingly, it sucks up the gametocytes of the Plasmodium. Within the gut of the mosquito, the gametocytes fuse. They basically form the zygote, which matures and migrates into the salivary gland of the mosquito. When the mosquito goes to feed again, the mosquito bites. Basically what it does, it injects an anticoagulant to be able to suck up the blood, and when it injects this anticoagulant, along with that, it injects the sporozoites of the Plasmodium, of the Plasmodium, and so those sporozoites end up in the bloodstream of the human that has been bit by the mosquito. They stay in the bloodstream very briefly because they very quickly migrate into the liver, and that's where they hide. Depending on the species, they hide for either 2 to 4 days or up to 10 days. The two different major human malarias, falciparum and vivax, have, in this case, they differ. Falciparum has a relatively short liver stage, while vivax has a longer liver stage, to the point that vivax can actually hide in the liver even for months, or there are some instances in which it seems it has hidden in there for years. So it's very difficult to target malaria when it's in the liver stage. But basically, while it's in the liver stage, it goes dormant for a little while, but then eventually it wakes up and it starts multiplying. And so the sporozoites mature into merozoites, they multiply, and then eventually they exit from the liver cells, they go back into the bloodstream, and this is what we call the erythrocytic cycle. They go into the bloodstream, they infect a red blood cell, they multiply into the red blood cell until they basically burst the red blood cell, and that is the symptomatic phase. That's when you start having the classic cycles of ups and downs of fever every basically 48 hours, roughly 48 to 36 hours. During this phase in which they are reproducing, the vast majority is basically the asexual cycle, but there are also some gametocytes that are being produced, and so then the next mosquito comes in, sucks up the gametocytes, the cycle continues. So that's the way the life cycle of Plasmodium works. So mostly what we are focused on is the erythrocytic cycle here and the pre-erythrocytic cycle in the liver. Not to target the Plasmodium inside the liver, but to try to prevent Plasmodium from entering the liver cell. Because like I said, once it's in the liver, it's very difficult to target. So this is a disease that has been around for a very long time. A lot of money is being poured into it, and clearly we're having an issue. We're still having an issue trying to get a handle on the spread of the disease. And so at this point, everything considered, the question is why? Why do we still have an issue? Why can't we figure out how to prevent this disease? And there are two very big categories of issues with their own complications that are basically stopping us from being able to eradicate malaria. One of them is drug resistance. Drug resistance obviously is familiar to everybody. We know it. Up here in green, you have basically, so you have years on the x-axis, and you have different drugs that have been used over the years, and when the green line fades is when resistance is detected at an appreciable level. And so as you can see, pretty much every single drug we have, including the combination therapies that we have now, they have some resistance associated with it. As I said, this is not, it's a known issue. It's happening with antibiotics, it's happening with antituberculars, it's basically happening with any drug we have. The question is, do we have a way of preventing it, or at the very least understanding how this drug resistance is evolving and what can we do about it? Now, most of the drug resistance usually starts on the boundary between Cambodia and Thailand. That's where it's the hotspot for drug resistance for a number of reasons. First of all, the incidence of the disease is very high in that area, but it's also because in that area, because of, I guess, the economic, socio-political structure of the area, it's many of the drugs that are available in that area are counterfeit, which means that they are at a much lower dosage than what they should be, which basically triggers the spread of antimalarial resistance, because the pathogen is exposed to a selective pressure that is not strong enough to kill it, but strong enough to select for the resistant types. And so from once they evolve, the resistance evolves here, of course it spreads in the rest of the world. Right now we are at the stage that we have seen a resistance to combination therapy for artemisinin of 4 different types of drugs. So up until relatively recently, this combination of 4 drugs worked, but resistance now has evolved for that. So now they're basically trying to figure out, okay, what do we do next in terms of getting a new set of combination drugs that will work. The other big issue in malaria is that basically we don't have a vaccine. And this goes to basically the prevention side. Now, the history of vaccines in malaria is actually kind of a curious history, because people have been going after a vaccine for malaria for many, many years. We're talking about basically at least 20 years here, possibly more. But no matter what we do, we can't find a vaccine that works. There are a number of them that have gone through, the classic phases. So you have your phase 1, phase 2, and phase 3 trials. And the only one that has currently been approved to actually go in the market and being used is RTSS. Now, the problem of RTSS is that as much as we would like to have a vaccine, it seems to be working almost exclusively in very young children. Excuse me, we're talking about children that are basically less than 2 years old, which leaves the other category, the 2 to 5 years old, very much exposed. And even in these children, it has an efficacy that goes between 26 and 39% at best. It starts by reducing malaria cases by 39%, but then after a few months that it's used, it basically goes down to 26%. So obviously it's better than nothing, but it's a far cry from being a really effective vaccine. Now, so at this point, the question is why can't we figure out a decent vaccine? That's pretty much the question that everybody is after. Now, if we look at RTSS, and we look at how it works, it's basically targeting this circumsporozoid protein. This is an alignment of different malaria species of the circumsporozoid protein. It's targeting the circumsporozoid protein that is necessary for P. falciparum to infect liver cells. And so it's basically attacking the pre-erythrocytic cycle, trying to stop Plasmodium falciparum from entering the liver. And if you look at the target of the vaccine, part of the target contains what is called a repetitive region. And these repetitive regions are known as low complexity regions that are basically areas within either protein or non-coding genomes in which there is one amino acid that is repeated many times, or maybe motifs of amino acids that are repeated many times, such as this case here. They are relatively short regions, but they are also highly variable. And so, and honestly, there are many different kinds of low complexity regions, and we really don't know much about them. We really know very, very little. What we know about these low complexity regions is that all the organisms, all the genomes that we have looked up until now have them. Whether they are pathogens or not, it doesn't matter. Every genome we have looked at has them, including prokaryotes. We also know that P. falciparum is one of the species that has the highest amount, along with the slime mold, has the highest amount of these low complexity regions in its genome. And so that's part of the reason why we are, in my lab, we're looking at low complexity regions in Plasmodium, because we have a lot of data to work with. But the other aspect that is interesting of low complexity regions is that if you look at the location of the low complexity region and you move basically farther upstream and downstream from the location, relative to how many SNPs, so how many mutations are accumulated, these regions here always have a high frequency of mutation accumulation, so they change quickly. And so the idea is, okay, these low complexity regions may be involved in some sort of adaptive response, whether it's for a pathogen or not, we don't know yet in Plasmodium. But we do know that in 2013, this paper was published looking at HIV, so completely different system, a viral system. And they looked in particular at the GP120, which is basically one of the genes that is involved in the pathogenicity of the virus. And this gene has 5 hypervariable regions, so regions that accumulate mutations a lot. And low complexity regions are present in 4 out of the 5. And these regions are the ones that allow the virus to escape the host immune system. So we are starting to see a connection, potentially, between these low complexity regions and the way the pathogen reacts to the host. So, what we are after are basically kind of 2 big questions. First of all, are low complexity regions functional? Do they actually do anything? Are they important for the survival of the organism itself? And by organism, I'm talking from the perspective of the Plasmodium. Because there is an ongoing debate. Some people say that they are actually neutral, they don't do anything, they are not important, and some people say they are functional, therefore they are under selection. If they are functional, how do we figure out if they are functional, and how do we figure out what they do? And so we go, from a genomics perspective, we go after 2 major things: sequence conservation and unique amino acid composition. Those are signatures, they are our proxy. It's our way of identifying if something is actually functionally important, because it should be conserved or it should be unique compared to the rest of the genome. And if they are functional, then what do they do? Are they involved in drug resistance? Are they involved in evading the host immune system? Are they involved in recognition of the host cells? What is it that they do? So this is what I was mentioning at the beginning. This is it's a very small tree of all the different, some of the major groups of Plasmodia. The 2 human malarias, falciparum and vivax, as you can see, are not closely related to each other. They actually belong to 2 different subgroups. In red you have the rodent malarias, and in black you have malarias that infect non-human primates, so chimps, gorillas, macaques, so on and so forth. So, based on the evolutionary history of the Plasmodium and on their genome, what we went after were signatures that we could identify. And the first thing we looked at was the amino acid composition of these low complexity regions. And what we saw is that this is Plasmodium, and this is what we call the CVK group, which include the vivax species. So, human malaria, and then we have 2 macaques malarias, and they have a strong preference for asparagine and lysine amino acids in the low complexity regions. But if you look at the distribution, it's the exact opposite. Plasmodium falciparum, super strong preference of asparagine in the low complexity regions compared to lysine. The CVK group is the exact opposite. It prefers to use lysine, which means that if we just focus on the yellow bars and the blue bars here, which are our human malarias, it's not host dependent. Meaning the way these low complexity regions are evolving doesn't seem to have anything to do with the host itself. It simply has to do with the genomic signature of the pathogen. So that's something that we didn't know that we wanted to keep an eye on. But then I also mentioned conservation and what we wanted to see if there are some of these regions that are conserved and some that are not. And we found that the majority, here we're focusing on the CVK group, the majority of the LCRs are not conserved, 72% of them are not conserved, but 28% of them are. And so we have two different signatures, some that are conserved within the whole CVK group, so they are conserved at this ancestral node here, which means this group, just to give you an idea, has been around for roughly 10 million years, give or take, so it's a very long time for these regions to remain conserved, and some that have been conserved more recently, just within the different strains of Plasmodium vivax. So we have, again, this different signature of conservation and non-conservation. Then, looking at both the conserved and the non-conserved, we looked at functional categories. We looked at the genes in which these low complexity regions are present, trying to figure out basically what do the genes do, and are they involved in any way in basically the potential pathogenicity of the disease. And what we have seen is that among the conserved LCRs, they tend to be enriched in transmembrane proteins, which are proteins that are supposed to communicate with the outside of the cell, and so potentially could be communicating with the host. And both the non-conserved and the conserved are enriched in adhesion and binding. And so there is this sort of signature of cell recognition and maybe host specificity that we are trying to go after. There is still a lot of work that needs to be done on this, but we are starting to see a little bit of a pattern. So, putting everything together at the very end, we were going after these two potential requirements to identify functional LCRs. We have found the sequence conservation, although not the majority, but we have also found the unique amino acid compositions. So from our perspective, LCRs, low complexity regions in Plasmodium, are functional, which means that they are under selection. We have not seen any host dependency in their composition, which, at least for now, is pointing us towards saying that they might not be involved in helping the pathogen evade the host immune response. We have found them, but we have found sequences that are variable quite a bit, which means that they could potentially be involved in drug resistance, in the evolution of drug resistance. This is where we're going to go after, trying to see if this variability is drug dependent, so that we can identify that signature. We have also found roughly 30% of them that have sequence conservation, which means that they could be involved in recognition and invasion of the host cell, for example, recognition of the red blood cell, so that the pathogen can invade it. And so we are starting to see a pattern here of low complexity regions being functional, potentially involved in cell-cell communication, and in rapid adaptive responses, which are the signatures that we are looking for in potential targets for future drug developments, future vaccine developments, so on and so forth. So that's where we are at with the genomics. of Plasmodium, looking at these low complexity regions. Of course, there are many other signatures in the genome of malaria that are interesting to go after. But we're hoping that putting together all these different stories, the individual mutations and the low complexity regions, we're going to be able to basically figure out how we can use the genome of malaria to go after prevention of the disease. All right, so I'll stop here. I just want to mention two of my students that did most of the work on this, Fabia and IINN, and then of course my collaborator at Temple that basically gave me all the genomes.